Hello students, welcome in the chapter Biomolecules. In this video we will discuss about the topic proteins and amino acids. Okay, now proteins are the polymers. Proteins are the polymers of alpha amino acids. Now polymers are such molecules which are made up of similar kind of units when they are repeated together. So such a molecule they are called polymers. As you can say, one chain is made up of small units when they are repeated with each other. So proteins are the polymers of alpha amino acids, and they are connected to each other. They are connected to each other by a bond called peptide bond or peptide linkage. Now what is this peptide linkage, what is this alpha amino acids, we'll see just now. So proteins are the polymers of alpha amino acids, and they are connected to each other by peptide linkage. A polypeptide, I can say a polypeptide with more than 100 amino acid residues, 100 amino acid residues having molecular mass more than or greater than or higher than 10,000 units is called a protein. Now this is the definition of the protein. Now let us try to understand the term what is alpha amino acid. Here if you see this picture, this diagram, you will see this is a molecule in which carbon is there in the center, and this carbon is bonded with carboxylic group at one end and amino group at another end, and with side it is having another carbon chain. This R may be a hydrogen, may be a carbon chain, maybe methyl, ethyl, or some other side chain, etc. And then there is hydrogen at top. Now see, if we have the traditional or common naming system, this carbon is bearing the functional group. So this carbon is the alpha carbon. Now alpha carbon bearing amino group is known as alpha amino acid. Okay, that's why this name alpha amino acid is there. The amino group is present on the alpha carbon, that is alpha amino acids. Now in this this is one you can say one example of amino acid, one example of amino acids. In this amino acids is a typical structure. There is a R. When this R will be differentiate, this R will have different values and different types of amino acid will be there. Today we are having 20 amino acids which are responsible for all types of proteins, whether it is animal protein, whether it is plant protein, or whether it is microbial protein. So these are different a different combination of R will make different amino acids, or total 20 amino acids are there which are responsible for the formation of these proteins. Okay. Now see, on the basis of this carboxylic group, the amino acid is having acidic nature, and basis of this amino group, amino acid is having basic nature. When both these are equal with each other, then the amino acid will be neutral. If basic group is more, then it will be the basic amino acid. If acidic group are more, then the amino acid will be said as acidic amino acid. Now see what is the polypeptide chain. When two amino acids they are combined together, here I will put NH2 group and here carboxylic acid. Okay. When these two one amino acids and then another amino acids, I will put NH2 here and then C H R and C double bond OH. Now when these two amino acids are combined together, here this amino acid will lose this OH minus group, this amino acid will lose this one of the H plus, and H plus and OH minus combined together will generate one water molecule, and the resultant structure will be NH2CH R C double bond. This is from one amino acid, that is on the left side, and NHCH R and C double bond OH. This is from another amino acid, you can say from the right hand sides, and here this bond, this attachment is called a peptide linkage or a peptide bond. Okay. Now when two such units are taken together, I'll say it is a dipeptide. When three such units will be taken together, it will be tripeptide. And when n such units will be taken together, in n such units, I can say here this H will be lost, here this O will be lost, and then we are having here and here, then n such units will be lost, and then the amino acid will be said as polypeptide. Okay, so when several polypeptides combine together, then the whole protein will be formed. Now this will be said as polypeptides, and at every step water will be lost, a condensation process is formed. In such way the proteins are formed by the combination of different amino acids. So in subsequent videos, in next part we will discuss further about these all amino acids and their types and their nature, etc. Now in a peptide linkage or peptide chain, we can see here this is another diagram. Here it is a peptide linkage. You can see this is one amino acid group, this is another amino acid group. Now here the OH part and the H part of this neighboring amino acid group will combine together and water will form. Here we can say when two amino acids are condensed together, then a peptide linkage is formed, CONH linkage. It is called a peptide bond or peptide linkage. Now two amino acids are joined together, that's why it is called a dipeptide. The combination of these two amino acids is a dipeptide. If three such units are joined together by peptide linkage, then we will say tripeptide, tetrapeptide, pentapeptide, and when many such units are formed, then the name polypeptide chain is there. And this polypeptide chain, when the mass is greater than 10,000 units, and in such combination 100 and more amino acid residue are combined, then the protein is formed. Now some more about this. Here you can say some other example of amino acids. Alanine is a neutral amino acid because the carboxylic acid unit, that is acidic unit or basic unit, are equal to each other. Here this is aspartic acid. This is the acidic amino acid. Why? Because it contains two carboxylate unit and one amine unit. Okay. This combination, when H of this carboxylic unit has been shifted, here the H from this carboxylic unit have been shifted to nitrogen of this amine unit, and then both end bear positive and negative part. Such unit is called a Zwitter ion. A Zwitter ion is the mutual transfer of H plus ion from one end of an amino acid to another, so that one end becomes negatively charged, another end becomes positively charged. Such a ion is called Zwitter ion. So it is a regular phenomena, the formation of Zwitter ion in an amino acid. So this is aspartic, that is acidic because it contains more carboxylic acid unit. Here it is the lysine, that is the basic amino acid, and the region here you can see only one amino carboxylic acid unit, but amino units are two. That's why it is basic amino acid. Here serine is alcoholic amino acid. Why? Because it also contains one OH group. Then cysteine is sulfur. Last year there was one question about this, the amino acid which contain sulfur units. So here it is SH, thiol group. That's why it is cysteine. And then phenylalanine, aromatic amino acid which contain one benzene ring in it. Okay, so these are some examples of the amino acids. Now we'll see another type of amino acids, that is essential and non-essential amino acids. Okay, essential and non-essential amino acids. Now the essential amino acids are those which cannot be synthesized, can't be synthesized in the body, can't be synthesized in the body of the human being, but to be taken in the form of a diet, are called essential amino acids. The example being valine and leucine. These have been asked several times in the competitive exams, or in competitive exams as well as in board exams. Now, non-essential amino acids, non-essential amino acids are those which can be synthesized, in the body, hence we need not to take them in the form of the dye. So we don't need them to be taken in the form of diet, are called non-essential amino acids. Here you can see the example: glycine and alanine. So this is the another category of classification of amino. Of deuterium, you can see the example: NH2 part, CH, R, C double bond, OH part. Now here this H plus part will be shifted from one end of the amino acid to the another end, and then we are having this kind of structure, this kind of dipolar structure, where one end is ammonium part and another end is carboxylate part. Now this dipolar formation is called adjunctive ion. Here you can see the list of 20 amino acids. Here in this, the name of the amino acid, full name, then the branch or the side chain or the R, then the 3-letter symbol, and then 1-letter code. So here we can see some are with stars and some others are without star. With star, these are the essential amino acids, and without star, these are the non-essential amino acids. So these are all the 20 amino acids you can see at a glance. Now, as far as the structure of protein is considered, four basic structure levels are assigned. Four structure basic levels are assigned to the proteins. And these are primary structure of protein, secondary structure of protein, tertiary structure of protein and the quaternary structure of protein. Quaternary structure of the proteins. Okay. Now, first we see what is the primary structure. Primary structure of proteins. It is the simple basic peptide sequence, the sequence in which amino acids are arranged in a polypeptide chain of proteins, is called or is known as primary structure of proteins. It generally has more than 100 amino acids. Generally has more than 100 amino acids in a polypeptide chain. Here you can see the structure of primary structure of protein. Here at one end you can say amine group, this is you can say N end, another end carboxylic group, so it is the C end. So this is the primary structure of protein. Here you can say the sequence of amino acids, okay, N terminal and C terminal. Here again you can say this is basic CHR and then this is amine group and this is C-double. So when such amino acids are combined in a linear sequence with more than 100 amino acids, then the primary structure of protein is formed. Now the secondary structure of protein is obtained when further coiling is done, then we are having secondary structure of protein. So here we can see in the secondary structure the folding of the linear chain, when the folding of the linear chain into specific coiled structure is done, then secondary structure of proteins are formed. There are three basic types: alpha helix, beta-pleated sheets, and then collagen helix. Here you can see the diagram. This is the alpha helix in the helical form, and then it is in the form of sheets, beta-pleated sheets, then it is the collagen helix. see another example, another diagram, secondary structure of proteins, and that is due to the hydrogen bonding between the different layers or the coils. Here you can say this is the alpha helical form, or this is the beta-Pleated sheets. Now how they are formed and what are their characters, we can say. First is the alpha helical form. So here we can say in the alpha helical form, it was given by Linus Pauling in 1951. Exists when R group is large. When the R group or alkyl group is large, then it is the right-handed screw with the NH group of each amino acid residue is hydrogen bonded to the carbonyl group of the adjacent turn. Here you can see in the diagram, here this is the hydrogen bonding NH of this and the carbonyl of another. You can say this is the dot 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 hydrogen bonding. Okay, also known as 3.613 helix. Since it is because in one turn the helix is approximately using 3.6 amino acids. Now if you see from here to there, in one turn there are 3.6 amino acids are being used. Okay, and that's why it is 3.6 helix. And the 13-member ring is formed by hydrogen bonding. Hydrogen bonding is there, and a ring with 13-member ring is formed. Carbonyl group and NH group of the peptide bonds are trans to each other. They are just in front of each other, opposite to each other, so that they can be hydrogen bonding. Now the another term is the beta-Pleated sheets. It exists when the R group is small. Due to the small R group, this beta-Pleated sheets is there, and in this conformation, all peptide chains are stretched out to nearly maximum extension and then laid side by side, which are held together by hydrogen bonding. Here you can say, so they are just side by side to each other. In this diagram you can say one, then the side by side to each other. So hydrogen bonding between this layer and this layer. Again here you can say the hydrogen bonding between here these two layers, you can say they are side by side to each other. So hydrogen bonding will be there. So one layer, then another layer. So it is the beta-Pleated structure of beta-Pleated sheets. So this is the secondary type of protein. Now here the second tertiary structure of the protein, and it is formed due to the further folding of secondary structure. Due to the further folding of secondary structure, we are having the tertiary structure. It can be further divided in two categories. When the folding is having a simple fiber-like structure, then it is fibrous protein. And if it is having a spherical shape, then we are having globular protein. And here we can see the diagram. This is the sequence of the secondary structure, which is further unfolding, and due to the extensive hydrogen bonding, they are combined to form the tertiary structure. Here you can see in this diagram the tertiary structure is there. So here you can see the tertiary structure. The helical polypeptide may fold upon itself and assume a complex but specific form. It may be spherical, then it is globular protein, or rod like, then it is the fibrous protein. Okay, and these geometrical shapes are known as the tertiary structure of a protein. Okay. So here we can say the two types and the difference between them, the globular protein and the fibrous protein. This is the globular protein and this is the fibrous protein. The point of difference between these, the structure result when the chain of the polypeptides, the structure result when the chain of the polypeptides coiled around to give a spherical shape. Okay, and fibrous protein, the protein chain are stretched out to the nearly maximum extension, so that they become longer in the form of fibers and then laid side by side, which are held together by intermolecular hydrogen bonds. Then the fibrous shape is formed. They are usually soluble in water and they are usually insoluble in water. This insolubility may be due to the extensive branching side by side, and there are some wonderful examples. Insulin and albumin are the common example of the globular protein, and examples are keratin, which is present, you can say, in hair and wool and silk, and myosin, that is present in our muscles. In our muscles we are having two types of fiber, that is actin and myosin. So myosin is that fibrous protein. Now NH group of each amino acid, that is the amino group residue, is hydrogen bonded to the carbonyl group of the adjacent turn of the helix, so that the extension of the hydrogen bonding, the capacity of the hydrogen bonding is further increased. And here the NH group of each amino acid residue is hydrogen bonded to the carbonyl group of the adjacent polypeptide chain. Okay, here it is hydrogen bonded to the adjacent turn of the helix, like this to the next turn, or here it is to the adjacent chain. Okay, adjacent polypeptide chain. The hydrogen bonding between these and hydrogen bonding between this and that. So these are the basic difference between globular protein, and it is asked in the exam several times. Here you can see all the four structures taken together. This is the primary structure, just a linear sequence of amino acids. Now on further folding or coiling, it forms secondary structure. This is the beta-Pleated seeds in the form of seeds, and here it is the second structure in the form of α-Helical structure. And on further coiling, this is the tertiary structure, which either may be globular or fibrous, and this is the Quarternery structures. Further extensive coiling of the tertiary structure leads to the Quarternery structure. Here you can see another example, the primary structure here, then this is the secondary structure. This is actually the α-Helical structure, and then further coiling of the tertiary structure, and here it is the Quarternery structure. Okay. Now in the Quarternery structure we can again say the Quarternery structures of the proteins. I can say when some structures, several tertiary structures taken together, then they form the Quarternery structures. Okay, or in some cases we can say some proteins are composed of two or more polypeptide chains. Okay, we can say two tertiary structures, and these two structures, they are subunits of that protein, and the special arrangement of these subunits with respect to each other is known as Quarternery structure of the protein. So these are the two or more tertiary structures taken together, then they form the Quarternery structure. Here it is the Quarternery structure. Okay, and the last important topic in this protein, that is denaturation of protein. This denaturation of protein is very important. Denaturation of proteins, because it has been asked several times in both exams. Denaturation means the nature, laws of nature, or I can say laws of the properties. So laws of the natural property of the protein is the denaturation of protein. So here we can say denaturation of protein. The laws of biological activity of protein. When a protein is in native form, is exposed to, is subjected to physical change, exposed to physical change like change in temperature or chemical change like change in pH. This is called denaturation of protein. Now coagulation of egg, here the temperature will be changed and then egg will lose its biological activity, it cannot be hatched then. And similarly curdling of milk, here pH is changed, so curd cannot be converted back to the milk, so its biological activity of the substance called milk is lost. So this kind of change is called denaturation of change, and it is irreversible. It cannot be reversed again. So this is another important topic, denaturation of protein. Okay. So now I hope you might have understood all these topics. So these are the topics, important points from the protein and amino acid part of the chapter biomolecules. Okay, and further we may discuss some questions related to that. Now let's see some questions which are based on the knowledge of your videos you've seen just now. The first question is the bond that determines the secondary structure of proteins. Out of these you have to find out the bond which is responsible for the secondary structure of protein. Second question, the sequence of amino acid in protein determine its primary structure, secondary structure, tertiary or structure, or none of these. Okay. Question number 3. The structure of protein arises due to folding, coiling and bending of polypeptides. Coiling and bending of the polypeptides chains, and producing three-dimensional structure is called. So it is primary structure, secondary structure, tertiary structure, or none of these. Question number 4. On hydrolysis, protein gives non-protein part and alpha amino acids. The non-protein part is called, see, the non-protein may be lipid, nucleic acid, phosphoric acid, or prosthetic group. Just think about this and search about this, you will find out. Okay, may not you find in this video, but you will find somewhere else. Just search on the Google or in the books. The type of protein consists of linear thread-like molecule which tends to lie side by side to form fibers are called globular protein, fibrous protein, conjugated protein, and none of this. We just discussed recently during the types of protein. You can see there. You can go back and see there and then find out the answer. The type of protein in which the molecules are folded together into compact units forming a spheroidal shape, spherical shape, are known as globular protein, fibrous protein, conjugated protein, or none of these. For a hint, I can say you insulin is the example of that. The function which is found in amino acids. So you can see whether this one, both A and B. Okay. So the functional group which is there. Now question number 8. What are the essential and non-essential amino acids? So we discussed recently, answer these with examples. Okay. Now define denaturation of proteins. I discussed just shortly back, so you can just go back and try to find out the answer. Name one amino acid which contains sulfur. Again you will find in the different types of amino acid in the example of that. So you can search the answer of these questions, and one answer I would like to give you. Question number 4. I think you may find difficult. So the on-hydrolysis protein gives non-protein part and amino acid, and that non-protein part which is associated with amino acid is prosthetic group. So this is the answer of question number 4. Rest of you can search in these videos and can answer them. I hope you got understand that. Wish you good luck for the exam.